ये और ये दोनों में। अरे तो कैसे होगा जी? मतलब नहीं पानी पीना कैसे ये वाला कैसे लाए ये स्क्रीन कैसे लाए तो वही ना आप दूसरा टैब खोले हैं नहीं प्रीवियस वाला कहाँ गया लाइक हम Yes.
Dr. Grisam, have you joined? Dr. Grisam, have you joined? Yes, yes, yes. Good afternoon, Grisam sir. Yes, Grisam sir. अभी चेहरा है अभी से शायद है Grisam sir. Should we start? Dr. Grisam. अभी तो बोले
Please start.
Yes, yes, yes. Now, can we start?
Yeah, yes, sir. Please start.
Yes. I think your mic is mute.
Yeah.
Okay, huh?
Yes.
Yes, Grisam ji, I'm Meenakshi here. Hello.
Yeah, sir.
Yeah.
Madam, please.
Yes. Should we start, sir?
Please start it.
Yes, sir. Respected Principal, Professor S.R. Padamdev, respected Professor Ellen Rai, Dean Faculty of Science, respected Professor S. Grisam, Department of Microbiology, Kakatiya University, Warangal, today's external examiner for the online PhD Viva Voce exam of Mrs. Amber Jabeen, Professor Virender Prasad, the internal examiner, the Professor Sanendu, Head Department of Botany, Patna Science College, all my distinguished colleagues, obedient students and friends. Sir, I welcome you to, on behalf of all my colleagues, to conduct the Viva Voce of Mrs. Amber Jabeen in online mode. Sir, with your permission, should I allow Amber to present her work? Should I allow her, sir?
Yes, sir. Yeah, madam.
Yes, sir. Thank you, sir. Amber.
Thank you, ma'am. Thank you, sir. A very warm welcome to Professor Grisham, sir.
Yeah.
My HOD, Nakshi ma'am, my internal supervisor, Dr. Virender Prasad, sir, and all the other professors present in this presentation, and all the participants, those who are present online. Thank you to everyone. So I am Jabeen, and here to present the role of harvest plants
Hello, Jabeen. Hello, Jabeen.
Hello,
Jabeen.
Your voice is loudly coming.
My voice is not clear.
So it is resounding.
Oh, there's echo in my voice, right?
Kindly all are mute your sounds. Please.
कर दिया ऑडियो बंद है
Please mute your mics, please.
Sure, sir. Am I clear, sir, now?
Now clear.
Okay, thank you, sir. So I'm continuing the presentation. The objective of my PhD work was to screen and quantify the phenolics in Phoenix dactylifera seed methanolic extract. Phoenix dactylifera is the scientific name of date palm, and the sample which I used was the seed. And the next objective was to evaluate the in vitro antioxidant activity of the Phoenix dactylifera seed and one of its active component, that was syringic acid. There are several active components, but I basically chose syringic acid as the active component of Phoenix dactylifera seed methanolic extract. The next objective was to assess the efficacy of both PDS and SA, that is syringic acid, on the health span and lifespan of animal model Caenorhabditis elegans. Another objective was to evaluate the in vivo antioxidant activity and lipid ameliorating effect of PDS and SA in the same animal organism. And the last objective was to untangle or delineate the longevity and health span in Caenorhabditis elegans signaling pathway. So this is basically my animal model. It's the figure is of Caenorhabditis elegans. It's a small microscopic worm. It's a free-living soil nematode, and the genome size is 97 megabase pair. It shows homology. About 83% of the human genes show homology to the C. elegans, and since it has a very short lifespan, high reproductive capacity, so it is very easy to manipulate in the lab. So that's why I chose this animal organism. C. elegans has found its use in various fields like diabetes, aging, cancer, neurodegenerative diseases, anti-inflammation, and obesity. And today I'm discussing it in the field of aging. This is the date seed, and the active component on which I studied was syringic acid. Date seeds are basically very rich in minerals and fibers, and the phenolic content in the date seed is also very high as compared to the pulp of the fruit. So that is why I used date seed. Okay. So this is the methodology which I adopted, like culturing of the worms and then preparation of the methanolic extract, and then analyzing the different phenolics through RP-HPLC, that is reverse phase high performance liquid chromatography. After the analysis and quantification of the different phenolics present in the methanolic extract of Phoenix dactylifera seed, I evaluated its in vitro antioxidant activity. Then its in vivo antioxidant activity was also analyzed. Its lifespan on the N2 worm. N2 worm is basically the wild type of strain of the worm. Its assessment on the lifespan was analyzed. Assessment of the health span, Lipofeusing accumulation, and stress tolerance was analyzed. Another methodology used was to analyze its in vivo antioxidant activity, where the elevation in the antioxidant activity was observed. And since antioxidant activity has a high activity on the lipid accumulation, so lipid accumulation was also analyzed. In this, we used a mutant worm called TUB1 mutant worms also. After analyzing all these effects, we finally came to delineate the pathway involved in the above mentioned processes using DAF16 null mutant. DAF16 is basically a transcription factor, a central regulatory transcription factor found in this organism. So I used the DAF16 null mutant worms, and to further confirm the results, I used GFP tagged DAF16 worms. So these were the results. Like the total phenolic component in the date seed was 449 milligram gallic acid equivalent. Total flavonoid was 458 milligram catechin equivalent, and the syringic acid concentration that was obtained through RP-HPLC method was 10.72 microgram per ml. Though the amount of the gallic acid was maximum, but I chose to work on syringic acid because the work on gallic acid was already established, while the work on syringic acid was never established before. So this is the first and the foremost work on syringic acid and also on Phoenix dactylifera seed on Caenorhabditis elegans. This is basically the chromatogram of Phoenix dactylifera crude extract of methanolic extract of Phoenix dactylifera seed. Here we can see there are 4 sharp peaks: number 1, number 2, number 3, and number 4. Number 1 peak coincides for gallic acid, which has a retention time of around 4 minutes. Number 2 peak shows the presence of paracumaric acid, which has a retention time of around 6 minutes. Number 3 peak denotes the phenolic of caffeic acid, and number 4 is the syringic acid, which is my active component, which has a retention time of around 19 minutes. So the concentration of the syringic acid in Phoenix dactylifera seed was found to be 10.72 microgram per ml. Now, after analyzing the different types of phenolics, phenolic compound in the seed extract, I did lifespan modulating effect of both the seed and the syringic acid. This is basically the survival curve of the lifespan. Here we can see that the mean lifespan of control worms where no treatment was given was 18.49 plus minus 0.66. And when the treatment of PDS at a low concentration of 250 microgram per ml was given, the mean lifespan increased to 21.91. That means there was an increment of 18.50. But as we moved to a higher concentration of 500, 1,000, and 2,000 microgram per ml, there was a significant decrease in the lifespan. So a lower concentration of the PDS seed provided a higher lifespan probability. So, and this is the same experiment but performed with another sample, that is the syringic acid. Here we see that the lifespan of the control worms where no other treatment was given was 20.14 days, while at a higher concentration of 600 microgram, which is the most effective dose of syringic acid, the lifespan the lifespan increased to 23.04. That accounts for a increase in mean lifespan by 14.40%. Now moving on to assess the health span, effect of PDS and SA on the health span, we use different parameters or the hallmarks of the health span. First was fecundity rate and pharyngeal popping rate. Fecundity rate is basically we observe the increment in the brood size or the reproductive capacity of the C. elegans. Next one was lipofusing accumulation. Lipofusing is basically an oxidized macromolecules that are resistant to degradation, so that gets accumulated as the organism ages. And as the organism ages, accumulation of lipofusing increases, which accounts for age-related problems also. And then another parameter for the health span was effect on stress tolerance. First is the fecundity rate. Here we observe that at a concentration of 500 microgram per ml PDS, the fecundity rate or the number of egg laying capacity increased by 117.9%, which is a very remarkable high increase. Similar observation was observed with syringic acid also. That is, but at a lower concentration of 200 microgram per ml syringic acid. There was an increment of egg laying capacity by 100.6%. This is the graph of the pharyngeal pumping. As the worm, in general, as the worm ages, there is a decrease in the pharyngeal pumping. And with the decrease in the pharyngeal pumping, there is a decrease in the calorie intake also. And this is an established fact that a decrease in the calorie intake leads to increase in the lifespan, which is known as caloric restriction. So it can be assumed that our treatment causes decrease in the calorie restriction that amounts to increase in lifespan. So in order to clarify whether the treatment causes decrease in pharyngeal pumping or the lifespan increment was due to the decrease in the pharyngeal pumping or due to some other reason. So we observe the pharyngeal pumping rate at the day of 5th day, 8th day, 10th day, and 12th day of the life of C. elegans. Here we observe that on all the days, the effect of treatment on the pharyngeal pumping was increased. Here, this is the dark blue is the PDS treatment and the pink one is of SA treatment. So at 250 microgram per ml PDS, the pharyngeal pumping increased by 92.84%, and at 600 microgram ml SA, the increase was 59.30%. So this result showed that there was no decrease in the pharyngeal pumping after the treatment of PDS and SA. That means there was no caloric restriction involved in the increment in the lifespan. Now the next slide shows the lipofeusing accumulation. The lipofeusing is basically an autofluorescent pigment that gets accumulated near the intestinal cavity of the worm. So this is the untreated worm. This is the adult untreated worm, which shows that the lipofeusing has accumulated all through the intestinal area. After the treatment with 250 microgram per ml PDS, the intensity of the pigmentation decreased. Here we can see that at certain places the pigment is very less. And with the same type of treatments with 600 microgram ml SA, the lipofeusing accumulation has also decreased. And this intensity of accumulation was quantified through ImageJ software. And this shows that there was a significant decline of 11.7% with PDS and 7.18% of SA. This shows that treatment with PDS or SA results in healthy aging. Now to see that the effect of PDS and SA provides, whether it provides resistance against different types of stress, we use different types of stress like thermal stress, where the worms were kept at a non-ambient temperature of 32 degrees centigrade for an interval of 2 hours. After 2 hours, we took the worm and kept at an ambient temperature of 20 degrees centigrade, and then again kept the worms for 2 hours at 32 degrees centigrade, and we counted the number of worms alive. So this is the graph of thermal stress. Similarly, H2O2 stress was given at a non-toxic concentration of 10 millimolar. So here also we observe that at a concentration of 250 microgram PDS per ml PDS and H2O2, the survival increase, there was a significant increase in the survival. Similar observation was made with syringic acid. Paraquat is also a type of chemical that causes oxidative stress. So with the treatment of the paraquat also, at 250 microgram per ml PDS concentration, there was an increase in the survival rate. And at 600 microgram per ml SA, there was also an increase in the survival rate of the worm as compared to the paraquat and methanol when given to the worms. So this experiment basically proves that the treatment of PDS and SA provided protective effect, or it provided indemnity against different types of stress, be it thermal stress or oxidative stress. Now moving on with the experiment, we did the in vivo antioxidant activity of the date seed and syringic acid. And through it, we showed that there was an increase in the catalase activity as there was an increase in the protein concentration. So as the protein concentration increased, the catalase activity also increased. This is the protein concentration, and this line shows the catalase activity. Similar observation was made with syringic acid also. And since there was an increase in catalase or any other antioxidant activity, so we hypothesized that there might be decrease in the lipolysis of the, there might be decrease in the lipid peroxidation of the membrane of the worms. So this is basically demonstrated by a decrease in MDA equivalent. MDA or malonaldehyde is basically produced when lipid peroxidation occurs. So when the worms were treated with 250 microgram per ml, there was a significant decrease in the production of MDA equivalent. That means there was a decrease in the lipid peroxidation of the worms. Similar observation was observed with syringic acid also. At 600 microgram per ml concentration, there was a decrease in the MDA equivalent generation. Moving on, we observed that there was the lipid ameliorating effect of PDS and SA also in wild type when the C. elegans were fed with high cholesterol diet. In C. elegans, there is no source of generation of cholesterol. So cholesterol is always incorporated through diet. So we can easily manipulate the amount of cholesterol generated within the C. elegans through external manipulation. So over here, we supplemented the worms with 20 ppm cholesterol so that enough cholesterol could be accumulated within the body. And then after the treatment with different concentration of PDS and syringic acid, we observed that the amount of the lipid accumulation or the cholesterol accumulation decreased, showing that there is lipid ameliorating effect of PDS. So this experiment was done basically quantitatively through Blig and Dyer method or sulfophosphovanillin reagent method. Same experiment was repeated qualitatively through two different types of lipophilic stain. One was oil red O stain. Here we observed that the control worms were stained very deep. After treatment, there was a decrement or lessening of the staining of the worms. But at a higher concentration of PDS, a reverse result was observed, where we saw that the concentration of the worms increased. This might be due to the higher amount of sugar present at higher concentration of PDS, or there might be some other unexplained reason. At a concentration of 600 microgram per ml SA, again there was a decrease in the staining of the worms. Same experiment was repeated with another stain, that is the Sudan black B stain, and similar observation was observed. This is basically the quantification of the images that we observed in these two slides. So this was done through ImageJ software, and we showed that at one particular concentration, the intensity of the stain decreased, both in syringic acid and Phoenix dactylifera seed extract. Now, after coming to all the conclusion that it provides certain indemnity against antioxidant activity, it also increases the health, indemnity against different types of stress, and it increases the health span and lifespan. We try to find and delineate the pathway involved. So we use DAF16 model of DAF16 model where we used DAF16 null mutants, and we also used DAF16 GFP tagged DAF16. This is basically the GFP tagged DAF16 image of control, that is the N2 worm. Here we see that the DAF16, that is the transcription factor, is equally dispersed in the cytoplasm. It can be translocated from the cytoplasm to the nucleus upon induction of any stress like heat stress. So when it gets translocated, the appearance appears to be a small dotted like appearance in the whole of the area. So this uniform appearance shows that it is localized in the cytoplasm. Upon treatment with PDS, there was no significant change in the appearance. Here also we see that the appearance is in the cytoplasm. Upon acid treatment also, the appearance came to be in the cytoplasm. That means there was no nuclear translocation from the cytoplasm to the nucleus of the DAF16. That means the DAF16 is might not be basically involved in this pathway. There might be some other pathway through which lifespan and health span extension was observed, which is a matter of further research. We also did experiment on the lifespan of DAF16 null mutant. DAF16 null mutant basically has a little shorter lifespan. And when we treated DAF16 null mutant with 250 microgram per ml PDS and 600 microgram per ml SA, we observed that there was similar increment in the lifespan of the worms as was observed in the wild variety N2 worms. So this shows that it is that the increment in the lifespan is independent of DAF16. Same type of experiment was repeated with thermotolerance of DAF16 mutants. A similar increase in the thermotolerance was observed in DAF16 mutants also as compared to the N2 mutants. So these experiments showed that DAF16 might not be directly involved in the extension of lifespan and health span of C. elegans. So this graph basically shows the role of TUF1 and DAF16 in PDS and SA-mediated lipid amelioration. TUF1 is basically a gene that is responsible for lipid accumulation in the worms. If there is a mutation in the TUF1 gene, it causes a lot of accumulation of the fat within the worms. So we wanted to observe whether this lipid ameliorating effect of PDS and SA is independent or dependent of TUF1. So here we saw that this is the N2, this is the wild type of a strain. Here this is the control, this is the treatment with the PDS, and this is the treatment with the SA. Lipid accumulation decreases to a certain extent. This is the TUF1 mutant worms. Here the concentration of the lipid accumulation is very high. Upon treatment, there is also a decline in the lipid accumulation concentration. And similar result was observed with SA also. With the DAF16 mutant also, similar result was observed. So we can also say that the accumulation of lipid or cholesterol is independent of TUF1 and DAF16. So it might be some dependent on some other pathway that is still unknown. So this was my inference. So finally, I would like to conclude the presentation. So the conclusion is that the Phoenix dactylifera seed extract and syringic acid prolong the mean lifespan of wild type C. elegans. PDS and SA treatment also provided indemnity against thermal and oxidative stress. And its supplementation promoted healthy aging marked by decrease in lipofeusing accumulation, increase in facility rate, and increase in pharyngeal pumping rate. The treatment with also decreased the total lipid accumulation, and the effect of increase in lifespan and increase in the health span was or might be independent of DAF16 gene and TUF1 gene that are basically involved in insulin signaling like pathway. It might be due to some other pathway that is still unknown, and that is a matter of further research. So this was the presentation, and this is the papers that I got published. Thank you. With this, I complete my presentation. Thank you, sir.
Sir, if you desire, you can ask a question.
So 2 or 3 questions are there, Jabeen.
Yes, sir.
One thing is that you are selected a palm seed.
Yes, sir.
So in that particular seed, more fats are present.
Yes,
That is
sir.
a basic character.
Yes, sir.
Why you are selecting methanol extract? Why not other extracts?
Because methanolic extract has the capacity to extract more of phytochemicals or secondary metabolites. Like
Yeah.
it won't extract the date. It won't extract the fat from the crude extract. Rather, it would extract the secondary metabolites and phenolics, like gallic acid or syringic acid.
Methanol is a high polar solvent.
Yes, sir.
If you are taking high polar solvents, the fats, other materials may be interfered.
Yes, sir.
Why not try for another solvent or one or two solvents to try to not to involvement of the fats and other materials? Why you are not to try for other solvent? Methanol is a good solvent.
Yes,
I
sir.
agreed. Really a wonderful solvent, high polar solvent. It brings whatever say the metabolic compounds, other compounds may be extracted, but the interference is more. For that purpose, which solvent is the best one?
Maybe DMSO?
No. So you go for a organic solvent. If you are trying for the organic solvents, methanol, chloroform or ethyl acetate, that you are not trying for that. So if
Yes.
you are going to ethyl acetate, you can eliminate the fats.
Okay, sir.
The second one is, are you try for the columns for separation of your biological activity purpose?
Sorry, sir, I didn't get you.
Are you try for the columns to purify the compounds?
Yes, sir. First, I tried column chromatography, and then after that, I did RPHPLC.
So in the column chromatography, what are the chemical compounds are interfered here? How many solvent fractions are you are taking place?
3 or 4 solvent fractions were observed, but I didn't carry out in the experiment through column
Which
chromatography.
solvent which solvent you are isolated this compound?
Methanol.
Again, you are methanol. If you are going for a methanol, entire compound may come out. Whatever extract you are taking in the field in the column chromatography, everything is coming out. Again, you are collecting crude. If you are going to the fractions of the solvents, so in the HPLC, that
Yes.
you are getting a last compound is your compound.
Yes, sir.
Then the other materials may be come out, and again a crude material is coming out. For solvent extraction purpose, column extraction purpose, though you must be select the solvent to isolate only pure compound.
Okay, sir.
So that should be identified. If you are going to publish a paper, so one confusion may be come out. Which solvent is purified solvent during the column chromatography? So that is one of the extension work you can done, please. Then,
Yes, sir.
are you find out any fats during the purification and all these things?
Any facts during the purification?
Yeah. It is very difficult.
I didn't work on the facts.
It is very difficult to isolate the fats from the generally seeds, particularly palm seeds. Are you find out any fats? If you are find it, so what type of experiment you are done for the identification of the fats?
I basically didn't work on the activity of the fats. I did on the phenolic compounds. So about the fats, I didn't go through that.
Acha, are you find out any methanol solvent in your HPLC experiment?
Yes, sir. Gallic acid, syringic acid, I use them as a standard with the methanol as the mobile phase.
Only methanol. So you are not shown a methanol peak there. Any peak is coming for the methanol?
No, I use a very high purity grade.
So here is
HPLC
that
grade,
you
so
are
there was no
keeping
peak for
that you
methanol.
are keeping here. So many peaks are available here, but methanol peak is not there. Generally, methanol character is always a debris are available in the crude.
Yes,
These
sir.
debris are toxic to the living any creature. So that's why I'm asking, during the extract, finalizing the compound, any fats are there or any methanol fraction is there. So I am asking, I'm doubt, that is a doubt for me. Any peaks are coming for methanol fraction.
These peaks were observed, so it might be the debris or the peaks of the methanol that wasn't studied. I basically use the standard of gallic acid and syringic acid, and through different research papers already published.
I'm not asking about your standard. I'm asking your crude, your extracted crude.
Yes, sir. This is the graph of the crude extract, where we observe these types of peaks that might be the impurity in that.
Another thing, last one is,
Yes, sir.
so before going to the publication, work is really wonderful, good.
Thank you, sir.
Before going to the publication, are you please plan it for the mass and IR for your extract material?
Yes, sir.
Because of everybody is asking about mass and this IR graphs. If you are find out the mass and other things, so it is whatever compound your compound isolated, that is clear. Under that can be find out your compound is very clean and purified.
Yes, sure, sir.
That one experiment, please extended it before going to the publication. Though you are writing of the thesis, so everything is good. Your introduction part is good. You are collecting more and more literature from the other parts of work. Then that your general this general discussion, material methods, everything is good. Though I am asking for some more extension work may done for your best papers, right? So
Sure,
your
sir.
thesis language and the typographical mistakes are not there. So it's a good. It is clearly described every chapter, right? Collection of the references, good, and the writing part is good. Overall, your contribution is good. So
Thank you, sir.
I am not seeing the other reports, but in my report. So I congratulate to you, and even Professor Birendra Prasad also. I am congratulating you. It's a very good attempt for the a plant extract for using the biological systems. Good. Congratulations, madam.
Thank you, sir. Thank you, sir, for your valuable comments. I would like to incorporate all these valuable comments in my further research work. And thank you, sir, for appreciating my work. And my supervisor also helped me a lot in arranging the thesis. Thank you, sir.
Good.
Anyone else wants to ask the question? Anyone else? Nobody is asking. Well, with your permission, should I congratulate her for the award of the degree?
Yeah.
Thank you, sir. Congratulations, number.
Thank you, ma'am.
Nandu ji.
Yeah. Professor S. Girisham,
Well, good afternoon, sir.
head and good afternoon, Professor. I am Arden and for your valuable time and your expertise to evaluate the thesis of the Patna University. I have gone through your biodata just now. I have seen we have completed many projects from UGC, CSIR,
Yes, sir.
AGCTE,
Yes.
and large number of publications. So, on behalf of department and on personal behalf, I extend you hearty thanks to evaluate the thesis of the Patna University. And I was just, I just joined your question answer session. I can very well see that how meticulous you were to examine the thesis, and you have gone through the thesis page by page, and you did not find any error in typing and other typological mistakes. And I'm also thank you to methanol used, what is the benefit of the methanol, what are the side effects of the methanol. So I find your personality that you are examiner and good. I am full in on behalf of the Department of Botany and that university works, and hope that in future also you will come with the
Definitely, sir.
I am
Definitely. Thank you. Thank you very much.
I also thank the program to have and bring and to researchers sitting in the room who have listened, though they did not ask any question. They should make the habit to be serious in the department in this presentation and have the meticulous observation, so that they should learn something if the good piece of work is being done. And in the last, I'm thankful to Professor Meenakshi Singh and other my colleagues sitting there, heading the department, will contribute to give time and their expertise to the progress of the department. In last, I am thankful to all who are directly or indirectly help the Mrs. Amber, Mr. or Miss Amber or Mrs. Amber said. So Mrs. Amber to continue this work and add the piece of good publication in the Patna University. This one. Lastly, I congratulate Mrs. Amber for their good work, wish and success in life. Thank you all.
Thank you, sir.
Thank you, sir.
Thank you, sir.
Thank you, madam. Thank you, ji.
We are leaving the.
Thank you, Professor Girisham.
Thank you, sir. Congratulations. A good piece of thesis that you are completed in your lab. So you are attempted a secondary metabolite isolation. It's a very, very important, sir. Because of nowadays everybody depends upon the plant extracts, plant metabolites. So that can be helpful for the living style of the everybody.
Thank you, and I am still remembering those days in Goa and Gwalior.
Yes, sir. Yes, sir.
Yes, with you for 15 days in both places.
Two
And
times,
myself
2 years.
attending a
Continuously 2 years.
refresher course
Oh, yeah, sir.
So,
Singaracharya,
okay,
myself.
sir, we will discuss about the sending of all the documents, reports and other
immediately I
things.
will send
Okay,
it, sir.
thank you.
Thank you, sir. Thank you.
Thank you, sir, once again.
Thank you. Thank you, madam. Congratulations, madam.
Thank you, sir.
Thank you.